Hey everyone, so we are starting a new unit on cell transport. So we're going to be talking about how the cell membrane actually is able to perform its function. So remember, the cell membrane is responsible for controlling what comes in and out of the cell. So the question we're asking is, how does the cell membrane control what comes in and out of the cell? Based off of that information, we can then go and look at two different cellular processes, one called photosynthesis and one called cellular respiration. Cellular respiration has to do with our respiratory system and our circulatory system. So we are going to be ending the unit with those two body systems as well. So let's just jump right into the content here. And of course, my computer is being slow now. So this specific video is going to be on, like I said, the cell membrane structure and its function. How does the cell membrane control what comes in and out of the cell? So this is going to be your reminder right here to make sure that you also have your vocab notebook open at the same time. And that way, as you are listening to this presentation, you can always pause and make sure you're also working on your vocab notebook to eventually complete it. All right, so for the structure of the cell membrane is made mostly out of three components. They are a phospholipid, a protein, and cholesterol. So the main structure is made out of phospholipids right here, and these are a lipid molecule, and they are able to create what is called a phospholipid bilayer. So bi means two, which means there's going to be two layers of phospholipids. So if you look right here, this is going to be an illustration of a phospholipid. It has what we call a polar head, and then it's going to have two non-polar tails right here. You don't have to worry about what polar means right now, but just know that when we take this phospholipid molecule, we put it together in this structure right here. So the tails will point in towards each other, the heads will point out either like away from the cell or in towards the cell. And this is how we create that lipid bilayer. So here is layer 1, and then here is layer 2. And so that's going to be the main structure of the cell membrane. You have this lipid bilayer surrounding the entire cell. Also keep in mind that all cell organelles are made out of this lipid bilayer. Now, as well as phospholipids, we also have different types of proteins. So in this picture right here, we have this type, here's another type of protein, third type of protein, and a fourth type of protein, this purple thing right here. Proteins are used for many different functions. Most of the time, it's either to communicate a message from outside the cell to inside the cell or vice versa. It's also used to help molecules pass through the cell membrane, acting as a tunnel for them to pass through that phospholipid bilayer without having to interact with those molecules. The third main component of the cell membrane is cholesterol. I know cholesterol gets a bad rap. If people's cholesterol is too high, they generally have to look at their diet, so what they're eating, as well as their exercise routine. Some people, it is more genetic, so it's going to happen to them regardless. In this case, cholesterol is a really, really good thing to have in your cell membrane. It is actually crucial that you have cholesterol in your cell membrane. So this little itty bitty thing right here is an example of cholesterol. And basically these are inserted every couple of phospholipids or so to help keep the membrane nice and fluid. So the idea is the membrane is constantly moving. You can almost think about it like the top of the water in like a pool. And so it's constantly moving and shifting, and you want to keep it nice and fluid. The cholesterol helps with that. Now, the proteins, they are also moving around on this surface of the pool, like a pool toy would, and they move around on the surface of the cell membrane, and they are constantly, like I said, in motion, moving around the cell. Come on now, there we go. So when we put these different components together, we get this model of a cell membrane called the fluid mosaic model. So we still have these three components. There are some other things in the cell membrane as well. But the idea is, like I said, this cell membrane is constantly in motion. The proteins, the cholesterol, constantly moving around the cell membrane, and the cholesterol is helping to keep the phospholipids nice and fluid. So this is where we get the fluid part of the fluid mosaic model. Mosaic is a type of art where you take a bunch of different little pieces from all these different places, put them together, and when you back up, you see like a big picture. And so the same idea here, we have a bunch of little pieces. We have a bunch of phospholipids, bunch of proteins, bunch of cholesterol. Like I said, there's some other things in there as well, but we take all these little pieces, put them together. When you back up, we get this great model of a cell membrane. So that's why we will refer to the cell membrane as a fluid mosaic model. So that's going to be the structure of the cell membrane, right? So it's made mostly out of a phospholipid bilayer. We have proteins in there to help with communication between other cells or within the cell, and we have cholesterol to help keep things nice and fluid. Now, in terms of talking about the function of the cell membrane, letting things in and out of the cell, we need to also keep in mind the structure. So the structure is going to lend itself to the ability to control what comes in and out of the cell. There are a couple of vocab words we need to go over first before we can talk about how these different molecules are able to either pass through the cell membrane or how they're not able to pass through the cell membrane. So this word here, hypertonic, these are all talking about the environment outside of the cell. So if you have a cell is sitting in a cup of water, the outside environment, the water, is what we are referring to with hypertonic, hypotonic, and isotonic. So hypertonic, very similar to hyperactive. It means that you have more of something. So this means there is more solute or a high solute outside of the cell, low solute inside of the cell, and we're going to see that water will eventually move out of the cell because of this. So solute right here is just the stuff that's dissolved into water. So if you take a cup of water and pour a bunch of sugar in there, mix it up, and let it dissolve, the sugar would be the solute in the water. Now, like I said, we're talking about a hypertonic environment. So maybe there's more sugar or more salt outside of the cell. And what happens is water wants to even that out, so it moves out of the cell. This is a picture example of it right here with a blood cell. So the cell will shrink up. It's very similar to when a grape turns into a raisin, the water like evaporates from the raisin, shrinks up, it shrivels. Hypotonic is the opposite. So this is when there is a low solute concentration outside of the cell, a high solute concentration inside of the cell. So water will move into the cell. That's this picture right here, and it will swell up. The same thing happens to you when you sit in a bath or a pool, lake, ocean, any of that, and you get the little like pruniness on your fingers. So those little like pruniness things on your fingers when you sit in water for too long is actually because you're sitting in a hypotonic environment. So water is going to move into your body, and those prunes are actually the skin on your fingers expanding to hold more water. If you want to do a fun experiment at home, you can sit in a bath for 20 minutes, however long it takes to prune up. But you want to weigh yourself before you get in the bath. And then when you get out of the bath, obviously like pat yourself dry. But if you weigh yourself again after the bath, you will actually see that there is an increase in weight because water has moved inside of your body. The last thing is an isotonic environment. This is when it's the same amount of solute in and out of the cell. And so the cell just stays how it's normally supposed to stay. Now, in the future, when I'm talking about the inside of the cell or the outside of the cell, I'm going to be using these words right here. So intracellular means inside the cell. Oops. Extracellular means outside of the cell. So I'm going to be using these words here to also help us explain where molecules are moving into or out of the cell. All right, so now that we have some vocabulary we can use to talk about how molecules are able to pass through the cell membrane, we're going to be talking about how these molecules actually pass through the cell membrane. So remember, keep in mind the structure, keep in mind the vocab. There are two types of transport to move molecules across the cell membrane. So molecules can move from the extracellular environment to the intracellular environment, from the outside to the inside, or molecules can move from the intracellular environment to the extracellular environment, from the inside to the outside. It can go in both directions. The first type of transport is called passive transport. So very similar, if you are being passive in your life, you're usually kind of like sitting around, laying down, you're not using too much energy. Passive transport, you are moving materials across a cell membrane without using any ATP, without using any of your cellular energy. So different types of passive transport are diffusion, osmosis, and facilitated diffusion. We will be talking about these on the next slide. The other type of transport is going to be active transport. And so similar to when you are active, you're up, you're using energy. Active transport is when you are moving materials across a cell membrane using ATP, so using cellular energy. And the types of transport for active transport are protein pumps, endocytosis, and exocytosis. And again, we'll be going into more detail in future slides. So these pictures down here are here to help illustrate these different types of transport. So for passive transport, remember there's no ATP required. So this type, diffusion, molecules can pass directly through the phospholipid bilayer. And then we have facilitated diffusion. I don't know why my mouse is going in and out. I apologize. Let's do it this way. There, that will help. So here we have facilitated diffusion. So this is showing that we are using some proteins inside the membrane to help move molecules through the cell membrane. It is not using any ATP in any of these examples. Over here, we have active transport. So this is showing, again, we are using a protein to move molecules across the cell membrane, but in order for this protein to work, we have to use ATP. And again, we will talk more about why these proteins are slightly different than these proteins in terms of their ATP usage. All right, so we're going back to passive transport. Like I said, we're going to be talking about passive transport in more detail. So the first type of passive transport we're going to be talking about is diffusion. Now, the scientific word for diffusion, my mouse is being super slow now. This isn't a good idea. Stop it. Okay. The scientific word for diffusion, or the scientific definition of diffusion, is the movement of a molecule across a cell membrane from a high concentration to a low concentration without using any ATP. Let's pause. I will explain this to you, okay? So let's say that you went to Moe's the other night, maybe like on a Monday, because Moe's Monday, they have the discount. Anyway, you go to Moe's and you get the giant homewrecker burrito. Like, I'm talking like stuffing this thing jammed packed. You got rice, you got cheese, you got the pinto beans, you got the black beans, you got chicken, you got steak, I haven't had a burrito in a while because I'm allergic to them. But what else do you put in there? Like tomatoes, you put the mushrooms, the peppers, the onions, you're putting the sour cream, the queso, the like hot peppers, the salsa, the mild salsa, the corn. You put all of it inside of there. It's like you need the 2 wraps to wrap up this burrito, like giant burrito, right? Anyway, you pay for your burrito, you go sit down, you eat the burrito in like 3 seconds flat. I know you were super hungry, right? So you go home, you're like, you know what? I'm feeling okay right now. I'm a little full, but I'm feeling okay. And then like a little bit later that night, you start to get a little bit of a stomach ache. So You go to bed early because you're not feeling that well. You wake up the next day, you're still kind of like the tummy rumbles. Y'all know what I'm talking about. You still got a little bit of those tummy rumbles, but you know what? You show up for school anyway. And I'm talking like the in-person type of school, not the virtual type of school. So we're all sitting in a classroom together, and you're feeling it now. You're feeling this burrito. So you're sitting there at your desk. You're like, oh man, I feel this thing. I feel this thing coming out right now. So you're gripping your desk. You got the white knuckles going on from gripping it so hard. You're like tapping your foot on the ground, trying to like distract yourself. And like this little trickle of sweat starts like pouring down your cheek just a little bit. And you just can't hold it in any longer. So we hear this giant we're not sure if it's wet or dry either. So in this case, right, we just farted. I'm talking about a huge fart. So the people that are closest to you are going to smell your fart first. And then as you start getting further and further away from you, those people are going to smell your fart second, third, and then the people the furthest away from you will smell your fart the last. That idea is the idea of diffusion. So we're going from an area of high concentration around your butt from where you farted, and those molecules will spread out or diffuse out across the room, and they're moving to areas of lower concentration as they spread out. So that is diffusion, right? Same idea if you're like in the hallway at school and someone's spraying their perfume or like Axe body spray or whatever, the people closest to them are going to have to smell that smell first, and then as it makes its way down the hallway, as it spreads out or diffuses out across the hallway, those people are going to smell it last. So that's what diffusion is talking about. Now, this picture right here is showing you how diffusion takes place. So this is our lipid bilayer right here. This is our cell membrane. So we have our little phospholipids. We have two layers of those phospholipids. So 1, 2, and then these molecules, or these like blue, what are they, hexagons? These blue hexagons are meant to represent different molecules. We can call it salt, we can call it sugar, whatever it is you want it to be. So in here, we're seeing an area of high concentration of these sugar molecules, right? This is the extracellular space. This is outside of the cell. So if we have a higher concentration outside of the cell, that means that this is a hypertonic environment for the cell. All right, so we have sugar molecules in the extracellular space. It is an area of high concentration. There's a lot of them. They are very close together. And so what happens is, in order to spread out, these sugar molecules are going to diffuse across the cell membrane into the intracellular space. So they're going to diffuse from outside the cell, across the cell membrane to inside the cell. And this process is going to keep happening until we have an equal concentration in the extracellular space and an equal concentration in the intracellular space. And that's what diffusion is. So at this point right here, once everything evens out, that's when diffusion stops. So molecules will still move across the cell membrane, but they'll do it at an equal rate. So the concentrations in the extracellular space and intracellular space will stay the same. Now, osmosis is the same exact thing as diffusion. The only difference is osmosis is only talking about water. So that example I gave you of sitting in the bathtub and letting the water like absorb into your body, water is diffusing into your body, and this would be an example of osmosis. You can do the same thing again. Take a raisin, put it in a cup of water. The water will diffuse into the raisin, which is another example of osmosis because, ready for this? So sugar can't actually move across the cell membrane the way we were talking about. Water can diffuse across the cell membrane. So if we have a little raisin in the water, it's filled with sugar. The sugar can't get in and out of the cell. So now we've created a hypotonic environment for the cell. So there's more water outside the cell than inside the cell. Water is going to diffuse into the cell. It's going to expand. Your raisin is going to turn back into a grape. Kind of. It's going to taste a little different, but you can still eat it. It'll still taste grapey. Anyway, let's talk about facilitated diffusion, okay? So same idea. We are still going from an area of high concentration to an area of low concentration across a cell membrane. We still do not need any energy. We do not need any ATP. However, these molecules need a tunnel to pass through the cell membrane. So an example of this might be sugar. It might be glucose, right? So basically all that's happening is the same idea that you saw over here. The only difference is they have to pass through the proper protein tunnel through the cell membrane. So I don't know if any of you out there have ever tried this. But what I do want to say is that if you are, no, I'm not going to say that. Anyway, so my question is, if you've ever tried this, like if you've taken a slug and you've poured salt on top of it, it will kill the slug. Like the slug will shrivel and die. So I want to try to explain to you why this takes place. Why does putting salt on a slug kill the slug? So let's think about this in terms of diffusion, right? Slugs have a lot of water inside of them compared to the outside environment. So they are sitting in a hypotonic environment. There's more solute outside, there's more salt, air, all that stuff. There's more solute outside of the slug than there is inside of the slug. There's a lot of water inside the slug. Now, when the salt is poured on the slug, we've created this hypertonic environment when we're talking about the salt. So if there's a lot of salt on the outside of the slug, there's a little bit of salt inside the slug, water is going to diffuse out of the slug. Now, when the water diffuses out of the slug, there's literally less stuff inside of the slug, so it's going to shrivel up. Also, slugs need that water inside of them in order to survive. So when they have no water, the slug just shrivels up and dies. And that's why putting salt on a slug will kill it due to osmosis and diffusion. All right, so let's talk about active transport. There's three types. There's the protein pump, endocytosis, and exocytosis. The protein pump is going to look very similar to facilitated diffusion. With a protein pump, usually what's happening is you are dealing with more than one molecule at a time for that specific protein pump. Also, instead of moving with the concentration gradient, so we have that like highly concentrated area of molecules, they want to spread out. That's what naturally happens. We are pumping molecules against the concentration gradient. So think about it this way, right? I'm sure a lot of you have a very messy room. You sort of just like leave your stuff everywhere. Everything is like littered on the floor, on the shelves, on your bureau, whatever is going on. Now, in order to clean your room, you pick everything up and you shove it into your closet. That's the same way the protein pump works. So we're taking all this stuff that's supposedly spread out and we are organizing it into one space. So it's moving against the concentration gradient. It's going in the opposite direction that it actually wants to move in. So again, in this picture right here, the sodium-potassium pump, this is actually how the axon of the neuron is able to pass that electrical signal down the axon from dendrites to terminal button. And we do this using sodium ions, so salt ions, and potassium ions. So what you see here is that we take our 3 sodium ions, they can bind to our protein. Right here where the little circles are, we give it some ATP, and we are able to pump the sodium ions outside of the cell. Now, while the protein pump is open on the outside of the cell, the potassium ions can come on in, and when the protein pump resets, it releases those potassium ions. So it's able to sort these different ions, these different molecules, where the body needs them, even though it's against their regular nature. The other two types of active transport are slightly different. They are called endocytosis and exocytosis. So remember, cyte, CYT, is a root. It is talking about the cell, which makes sense. We're talking about cell transport. Endo means to enter, to move inside of. Exo means to exit, to move outside of. So endocytosis is moving materials into the cell. This does require ATP because it's active transport. Exocytosis is moving things out of the cell or exiting the cell. Again, requires ATP because this is active transport. So these pictures down here are meant to represent endo and exocytosis. So what you can see is this blue line here is meant to represent your cell membrane. So extracellular space is blue, intracellular space is yellow. Molecules will, sort themselves into this space that the membrane is creating. It's kind of like enveloping these molecules. And eventually the membrane will pinch off to form a vesicle inside the cell. And so we were able to move materials into the cell. Endocytosis. Exocytosis is basically the same thing, just in the reverse order. So we start with a vesicle with molecules inside. It binds to the cell membrane and kind of like opens itself up to release the molecules outside of the cell into the extracellular space. So I get that this is probably hard to visualize. I have two videos I want to show you. This first one is endocytosis. I'll explain the second one when we go to watch it. So what we have right here is an amoeba. This is a single-celled organism. This amoeba is going to use endocytosis to eat this single-celled organism over here called a paramecium. There we go. All right, so here is our amoeba. My mouse will move. Here's our amoeba. The amoeba is going to extend its cell membrane right here to envelop the paramecium into a vesicle so that it can eat it. All right, so the paramecium is basically trapped at this point. You'll see it flailing around, but the amoeba right here is going to start to bring its cell membranes together in order to pinch off to form that vesicle that is inside of the amoeba. Should have played this in like 2 times speed or something. All right, so you're seeing the membrane pinch off right here. There it goes. We now have that lipid bilayer, that phospholipid bilayer around this vesicle right here, and we've rejoined our lipid bilayer around the outside of the amoeba. Here's the next video. This is showing you that sodium-potassium pump, that protein pump that is the active transport. So they are going to explain it to you in this video as they show it to you. It's a dry video, but the explanation with the visual is very good.
Active transport is a type of cell transport that requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules or ions to move from an area of low concentration to an area of high concentration. This is commonly referred to as against the concentration gradient. One of the more common examples of active transport is the sodium-potassium pump. The job of the sodium-potassium pump is to move sodium ions, or Na+, out of the cell, and potassium ions, or K+, into the cell. Let's take a closer look at how this happens. The sodium-potassium pump has binding sites for 3 sodium and 2 potassium ions. Let's follow the movement of the sodium ions first. After 3 sodium ions are positioned within the carrier protein, an ATP molecule is split, releasing phosphate. This phosphate binds to a location on the exterior of the carrier protein, causing the protein to change shape. As the protein changes shape, the 3 sodium ions are released to the other side of the membrane. Next, 2 potassium ions position themselves within the carrier protein, causing it to undergo another change in shape. In the process, the phosphate molecule is released. Once the phosphate molecule is released, the carrier protein expels the potassium ions into the interior of the cell. The carrier then resumes its initial shape, completing the cycle. Notice that for every 3 sodium ions leaving the cell, 2 potassium ions enter. Since both sodium and potassium ions have a positive charge, this unequal movement causes an electrical gradient to develop across the plasma membrane of the cell. A number of cellular processes, including the generation of nerve impulses, use this electrical gradient.
All right, so you can always check the slide deck if you want to re-watch any of these videos. At this point, I'm going to stop this particular video here. Please check in with your vocab notebook. Make sure that you have filled in the vocab. Again, open up your slide deck that goes along with this and just click through the slide deck to make sure that you have all your vocab words and that you have found all the definitions for your vocab words as well.